The IRMACS Centre is proud to present the SFU Fellows of the Royal Society of Canada Seminar Series. This bi-weekly series hosts 5 presentations per semester. For the spring 2013 semester, the presenters belong to the Departments of Economics, Physics, Humanities, and the Faculty of Education. Today's speaker is Dr. Martin Zuckerman from the Department of Physics.
Our today's speaker is Professor Martin Zuckerman, and it is my pleasure to introduce Martin, who's been a long-term project leader here at IRMACS Center. He is from the Department of Physics. Martin did his undergraduate and graduate work at Oxford University, completing a BA in mathematics and DPhil in theoretical physics. After 1 year as a postdoctoral fellow at the University of Chicago, he taught for short periods at the University of Virginia and Imperial College, before moving to McGill University in 1969, where he remained until he retired as William C. McDonald Emeritus Professor of Physics in 1999. In mid-20s, he joined the physics department at SFU, where he remains a very active member of the soft and biological physics group. Early in his career, Martin worked in solid state and many body physics, but spent the bulk of his career working in theoretical, biological, and soft condensed matter physics. He is the author of more than 200 papers, has an H index of 41, and besides being a fellow of the Royal Society of Canada, he is an international member of the Royal Danish Academy of Science and Letters. He was a founder and the first director of the Center for Physics of Materials at McGill University from 1989 to 1999. The title of his talk today is Novel Synthetic Nanomotors that Mimic Biological Motor Properties, a Wonderful Activity for Retired Researchers. So please welcome Professor Zuckerman.
Thank you very, very much for coming. I'm very flattered. Also, I don't quite know what 41H means, but it sounds good. You know the title, so I'll start straight away. I'm going to start looking at macroscopic transport versus molecular transport in cells. I'm then going to continue with the properties of a particular cytoskeletal motor protein called kinesin. I then go on to look at our synthetic protein-based motors, our various designs, and the motivation. We'd like to employ a modular protein design route using operating principles of biological molecular motors to construct more controllable and useful synthetic nanomotors. And specifics, design and construction of synthetic protein motors that are capable of directional motion along a track using only combined non-motor protein and DNA parts. I just wish to remind you that molecular motors are protein motors, and that's why we chose to use protein moieties to construct them. Okay, to go on. Suppose you want macroscopic transport and you want to transport some apples. Well, you could sit under a tree and use Newton's laws and wait for them to fall. But you're much better off putting them in a cart or putting them in a truck. Let's see. And but the transport of material as such requires a machine which functions using fuel in the macroscopic world. For the horse, it's food, metabolic energy, i.e. chemical energy, eventually that becomes mechanical work. Same for the truck. Petrol, I'm sorry I used the English word, didn't want to use gas. For an aged person, that might mean something else. Petrol, internal combustion to mechanical work, and the laws of motion of the cart and the truck. The motion of the cart and the truck is directional and can be fully determined by Newton's laws of motion, I hope, i.e. F equals ma, as you know. Now, I just want to remind you that here we have a cab which is pulling something, and which is pulling a load, and that's exactly what we end up with for the cytoskeletal motors, or some of them. So, then we have, the important thing is that small fluctuations in temperature obviously don't concern you in this case. Huge ones may. So, now suppose you're looking at the transport of small molecules in water in an aqueous medium. If they're small enough, then you're in the overdamped limit. So acceleration doesn't count. Newton's laws are negligible, even though they're there. And what you have is if you apply a force, the force is against the hydrodynamics, a drag coefficient, so you end up eventually, if it's a constant force, with the force over the drag coefficient, which is proportional to the viscosity, i.e. the friction in the medium. Now suppose you just put a molecule, a simple molecule, and a small molecule, you want it to get somewhere in a cell. Well, one thing you might hope is that at least it would be able to do random motion, i.e. it would do a random walk, which I'm sure you're all familiar with, i.e. the walk of a drunken man, which is caused by the water molecules scattering against your small molecule, which you assume rather bigger. This has no directionality. Basically, go back on that one. So basically what you have is, in this case, you have, instead of a constant force, you have the scattering of the water molecules gives you a random force here, and the random force and the motion are due to temperature fluctuations, and it's slow, i.e. the distance, there's no directionality, and the distance is of the order of time to the half, whereas the time taken to the half, whereas if it were directional, it would be proportional to the time taken. Well, now you have another problem. Not only do you have this situation, but you actually have your molecule in a crowded cell. So you have this very crowded cell with all these fibrils, and so you're even worse off. Though I did read an article which said that it may be evolutionarily useful, and that is diffusion in a crowded environment, i.e. the cell interior, very crowded. Then you have subdiffusion, which is even lower. So suppose you want to transport a set of molecules, such as, for example, a set of some neurotransmitters in a vesicle. In your crowded environment, you're not going to do anything if you just leave it there and hope it'll diffuse. So nature's answer for fast directed molecular transport inside a cell is that you basically evolution has constructed rather beautifully molecular nanomotors moving on a track. So let's have a look. This is thanks to Nancy. We're going to look at a particular motor. This motor is called kinesin. There are 45 different types of kinesin, moving different things, and they walk on a microtubule. The tendency is to go from one cell to the other, from one part of the cell to the other, particularly the area where the production area to the use area, if you like. And so you have them moving along a track, and I'll mention the speed later on. And so you can see here, you have, this is a particular, in this case, well, this is a vesicle actually, so you have a motor moving a vesicle from the negative to the positive end. And you have one type of motor called kinesin, which I'll concentrate on, that moves from the negative to the positive end of microtubules, and you have another motor which can move from the positive end to the negative end. And if you stick both of those, as I shall show, on a vesicle, say, on a bag with stuff in it, then you can use one to move one way to deliver, and the other to move the other way to go back to the nucleus. So you have these various roads here. You also have if you look at this, you have, I'll look at the structure in a moment. What you have are two, what are called motor heads. I'm sorry about the word leg here. And you have a stalk here, and you can carry a cargo. Cargo is usually huge. Here you have 50 nanometers, but the cargo is micrometers. So it's very much like the picture of the cab and the truck that I showed you. And even more, the sizes are even more extreme. So the cargo, well, kinesin, particularly in neurons, it can, well, in this case, it can move actually organelles like mitochondria, or the important things are sacs of chemicals, i.e. lipid vesicles filled, for example, in neuronal cells with neurotransmitters, which are created in the soma or the area of the nucleus. These are micrometers in dimension. So if you actually look at kinesin, what you have, kinesin doesn't walk on its feet, it walks on its heads. And I always have problems writing papers. I wrote a paper saying we have a motor that whose feet are, and they say, no, they're not feet, they're heads. So there are problems. Still, what you have is you have two heads. This is where the catalysis occurs, which I'll mention in a moment. That's where the actual enzymatic reaction happens, and I'll mention in a moment what it is. I think you all know it. Then you have two what are called neck linkers. They're linkers here. Then you have the stalk, and you have the cargo on the end of the stalk. And if you want to look at, if you want to have a look at what it looks like, because this is a protein, you can see the two motor heads here with the catalytic sites with the actual binding sites where the fuel goes. And here, these small things here are the neck linkers, and that's the stalk. Okay, next slide. Okay, the motors are protein motors. They're what we call mechanoenzymes. That means they use a catalytic process to convert chemical energy into mechanical energy, and they use the hydrolysis of ATP as the energy source. They move directionally along an actin, or in my case, it's going to be a microtubule track in an aqueous environment, which is usually crowded, as I said. The interesting thing is they're strongly affected by thermal fluctuations, as opposed to the macroscopic world, which act like a tempest. They use these thermal fluctuations to produce directed diffusion, which is known as thermal ratchet motion. And what I want to do now is just show you an enzymatic cycle. So what you have here is you have these two heads here, and I've only got the two neck linkers. This is a very simple scheme. So you start off with the lagging. Assume the leading head is fixed. You look at the lagging head. It's already bound ATP, and the lagging head hydrolyzes ATP to ADP plus PI. The Gibbs free energy that comes from that causes a change in conformation, and the only change in conformation I've shown here is that, that causes a change in conformation. In particular, the leading neck linker is strained, and then as soon as the phosphate, you've got ADP goes to ADP plus phosphate. Then the next step is that the phosphate is released, and in releasing the phosphate, you also relieve the tension, and the tension causes something called a power stroke, in which, and it also, and you also at the same time get unbinding by the lagging head. So you have unbinding and a relief of tension, and the relief of tension is supposed to take the lagging head over to the leading head side, and that's a bit like a swish, which is called a power stroke. And then the next thing is that once it gets to the leading side, it binds, the ADP comes off and it stays there, and then the whole process starts again. Now, so I have the new leading head binds to track and releases ADP. I've put this arrow here to say this is not the whole story, and in fact, the rest of the story is rather interesting. So, this is the rest of the story. Basically, the rest of the story here is that here you have the process I mentioned, then you have the power stroke. In the power stroke, you notice, first of all, the neck linker is extended, but at the same time, there's still energy left to change the conformation of the leading head. And what happens in the leading head is that a cleft opens, and part of this neck linker zippers onto the leading head, as you can see here, zippers onto the leading head, shortens it in such a way that the diffusional search after the power stroke has been done is restricted. So you get a restricted diffusional search, which really puts the head very close to the binding site, and then it binds. So you have zippering of neck linker onto motor head just before binding to microtubule. This turns out to be a three-step process, basically based on electrostatics. and it leads to restricted diffusional search. So the diffusional search is restricted and binding to track. Okay, I just want to show that as a movie while I go on. Hopefully, yep, sorry, what do I do? So it's just going backwards now. Now you see the whole process, which I won't describe again. Now the interesting thing about this motor is that it can take more than, that's kinesin, can take more than 100 steps of 8 nanometers each. There you can see the neck linker binding. You'll see it again next time. Can take 100 steps of 8 nanometers per second at speeds of 1 micrometer per second. That's equivalent to a sprinter running 100 meters in less than a second. So it's really quite tough. Then each step uses one ATP molecule, that's the fuel in the cell. And as I said, that's coupled to directional motion, power stroke plus directed diffusion via conformational changes. The efficiency, well, there's a big argument about efficiency. You know, some people say 50%, some people say 100%, but I don't want to get into it. Yes?
In the animation, am I noticing that one of them goes clockwise and the other one?
Yes, well, it's what's called a hand-over-hand mechanism. Because there was an argument. Some people thought it was an inchworm mechanism, and then Yildiz, I think Yildiz was a young Turkish guy working in a group, did very well, made pointed, decreased the fluorescence, decreased the fluorescence beam, if you like, and he was able to see it was a hand-over-hand mechanism. So it really sort of goes like this, you know. I'll show you a picture of what happens with the stem next. I think the answer is yes, but the stem is very long. So I don't think so. Okay, let's see. So here we have a movie of what happens in the cell, and I should remind you that it's a very nice movie. It's the same thing. I think it's by the same group. And what you see is it moving there, but it's supposed to be moving in a crowded environment, but that crowded environment is not crowded enough. So it's probably much worse. Okay, that's kinesin, and I'm just going to check the time. That's not too bad. That's kinesin, and I just wanted to introduce the aspect of the motion of kinesin, which is very processive, i.e. it sticks to the track for 100 steps of 8 nanometers each. And I wanted to show you the detailed motion, which I will be using. I'll be trying to mimic. And that's the whole idea of showing you this. Okay, now we want to build some molecular motors. This is the happy crew that of whom I've had the pleasure of working with. They're all their names. They're the principal names here, including our own Nancy Ford, who's been a fantastic colleague. They're all fantastic colleagues, actually. And the research goals are, basically, it's my side is the simulation, determine design criteria for synthetic bio-based nanoscale machines, basically, and also apply these criteria to experimentally synthesize and characterize novel protein-based molecular motors. That's just to remind you. So that's what the next section is going to be about. Great. So, this is Nancy Ford's slide. She's been a real help. I'm such a bad typist that I'm surprised I got anything down at all. So what are the requirements for a synthetic motor? A motor that you're actually taking protein parts, putting them together. What do you need? One of the things is you need it to be unidirectional, that it walks preferentially on a track in the forward direction, whatever you choose the forward direction to be. Able to take many steps before detaching, that's processive, ideally walks quickly. But as Nancy points out, sticky feet, if it really sticks very well, so that it's very processive, you have very good binding, that could actually make it slower. Then, the other thing that's very important is it's the same, I mean, suppose you have your truck that we started with, and you just try to pull it backwards. Well, if you weakly pull it backwards and you're a human being, or a goat or something, you'll be pulled you'll be pulled along. So basically the truck can still drive forwards even if you're applying a backward force, but eventually you can have a force strong enough that will stall it and maybe even reverse it. Obviously you'd need another truck to do that. So this is the same with these motors. We want them to walk forward even if pulled backward. And this is the clearest indication that the walker is a motor. It's actually producing motive force. So this has the ability to perform mechanical work with some energy input. And I'll talk about the energy input. So this is our first motor. Let me see if I can... I've called it our original nanomotor concept. And we've called that the tumbleweed because it acts like a tumbleweed. I used to have a picture of a tumbleweed. I'm sorry I don't. So let me tell you what we do is we take protein parts to like Lego. We take little protein parts and put it together. The only difference with Lego, of course, is that the pieces stay the same in Lego, whereas when you do your molecular biology, which is so nicely, I did a lot of this work in Australia, which is so nicely called in Australia molly-bolly. So when you do your molly-bolly, you didn't even get a laugh. Wow. Sorry, mate. So, nice place, Australia, by the way. So when you actually do the molecular biology, you can actually change some conformations and so on. Well, let me tell you what the motor is. So I start off with 3 ligand-gated dimeric repressor proteins. And I'll tell you what a repressor is in a moment. It's not psychological. Repressor proteins labeled RA, RB, RC, and they're connected by coiled coils arranged in a flexible Y-shaped hub. So I have this hub here. Here I'm talking about roughly 5 to 10 nanometers maybe. And if you look at this hub, basically, even though the movie I'm going to show is not flexible, these 3 things are flexible here. So it can, apart from certain steric restrictions, they're just tiny, tiny oligomers. And that's one of the problems in construction. Then you have this hub with 3 stiff coiled coils, so these are totally stiff. And then you again have flexible linkage to the dimer, which is the repressor protein. And the important thing here is that you have 3 different repressor proteins. And I've labeled them RA, RB, RC. And I will apologize in advance for the next slide because this notation may change. So, and I was very careless about that. So, what's the next thing? Now, the RA, RB, and RC are able to bind to corresponding recognition binding sequences incorporated in a dsDNA track. So here's your track, and here I have a binding sequence A, or recognition sequence A, they're palindromic, recognition sequence B, and you can see that here I already have these bound. Now why do they bind? Well, that's the next step. The binding sites are arranged I first of all, in order to make the motor work, arrange the binding sites in a spatial asymmetric periodic sequence. ABC, 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 etc. So it's basically a one-dimensional lattice with a symmetry. And what happens? The repressor proteins can only bind in the presence of corresponding ligands, labeled ABC. So what happens is, if you have a repressor protein like this, where there's no ligand bound to it, ligand being a small molecule specific to the protein, if there's no ligand bound to it, I mean, it just sits in this conformation. As soon as ligand A, I've called these ligands A, B, and C, ligand A binds to, as soon as ligand A binds to the repressor A, then the repressor A changes conformation and can bind to the appropriate point in the track. The way that we want it is that this repressor only binds here, this repressor only binds here, and upstream there'll be C where the other repressor can bind. So that's the story. Here I've got the ligands in solution, and I'm just showing that the ligands can, even though I don't have the ligands probably somewhere there, you can't really see it. So that's the mechanism. So how do we make it move? Well, the motion of the motor is controlled by a microfluidic supply of ligand pulses. So you keep pulsing in ligands, and the way that you do it, you first of all, you do this in a time periodic sequence. And let's start, suppose we have A and B in the, aqueous medium around the motor. then you can, A will allow A to bind, B will allow B to bind, and it has to bind A here and B here because this is the way we've controlled the periodic sequence. What we then do, and I'll show you a movie of this because this is not my main motor, what we then do is we remove A and put in C. We put in ligand C, which is related to this protein. And so what happens is that as soon as we do that, because we have no more A is removed, this one unbinds, this one stays bound, and the whole thing starts moving. But we've so arranged the system so that RC can't bind backwards, it can only bind forwards. So we have to make this pulse long enough so it can bind forwards. Once it's bound, then we again change the ligand atmosphere, keeping C fixed, and then B will start. Then the whole thing will start moving, and it'll go forward, and so on. So what I want to do is I want to show you I was told that I had to tell you this. So repressors are proteins which inhibit transcription when bound to DNA. I really have to tell you that, otherwise. Yes?
Are there any motors in nature that...
No. Well, there are DNA. I don't know of any. There are actually a lot of DNA motors, which were, there's a big construction by Turberfield and other people of DNA motors walking on DNA. But as far as I know, there's not even a concept of a protein motor working on DNA. And I'd like to know some information. Maybe somebody can tell me. Oh, well, of course there are motors in DNA itself, opening up DNA and like helicases and so on. Polymerases, polymerases. Yeah, the answer is yes. You have DNA polymerase, RNA polymerase, helicase, and others. Sorry about that. Just shows you I'm a pretty lousy biologist, as my colleagues will agree. Thank you for the questions. So, what I do is I make a very simple model, which I shall show you in a minute. I use what's called Langevin dynamics. You can see here, oops, sorry. This bit is F equals ma. We don't need that. So all we have is what I showed you to start with. That's the velocity. Here's whatever internal forces and external forces we have, conservative forces, together with a drag coefficient, and then we also add to this force. So this is a force balance equation, velocity force balance, it's a force balance equation actually. We have the drag coefficient here, and here we have, for the thermal fluctuations, we have a random force, and you can see that when you discretize this, you get something that looks like that, and the random force comes, the temperature comes in from the correlations between in the random force. So basically what we do is we use this, and we just use this to do the simulation. I should mention that this is very simple hydrodynamics. We actually have no interactions, hydrodynamic interactions between our various parts of the motor. So this is the simplest approximation, but seeing the motor small, it probably works quite well. If it were a huge polymer, it probably wouldn't. So what I've got here is, it's not quite the motor I presented, because I started off having something that I called the Y motor that was totally stiff, i.e. all the angles are fixed. Since then, I've done better than that, but the movie shows this. So this roughly shows you what goes on in an ideal case. So the motor first has to bind. So in the first pulse, it's binding, say, A and B. It looks around, does a diffusional search. It's sort of in the medium, and it binds to the track. So you can see A and B bound there. Then it's fiddling around until the end of the pulse. And then the end of the pulse happens, it goes all over the place. Well, actually, it hasn't, the end of the pulse hasn't happened. Come on, end pulse, please. Gosh. I hope this is working. Let's try again. It did switch. Oh, good. Let me just go back. Okay, yes, it switches quickly, actually. Let's do it again. This bit is better. That shows you the random motion. Then I'll have to look for the switch. I always have to. My brother-in-law did this very sophisticatedly. Switches very quickly, actually. Yeah, now it's switching. There. So it basically keeps going along the track, like a tumbleweed. Good. Well, the directional driving force is both the asymmetry of the fuel and the asymmetry of the track. So it's basically the fuel here is the chemical potential of the ligands, because they're being changed all the time. Also, I should mention honestly that in this very simple simulation, I assume that I have in my pulses of ligands, I assume that as soon as the ligands are in the atmosphere, they bind, and so they bind to the repressors, so the repressors can bind to the track. And then as soon as I stop, they unbind. So this is a very brutal approximation, which gives you an idea of how things work. We have improved on it using a master equation. Pardon? Oh, the energy here is, I think I have that on another slide. I have to, it's quite small, and it's roughly of the order of something like ATP. I hope I have that on another slide. Okay, so the idea of the tumbleweed motion here is it exhibits processive motion via directed diffusion, track binding, and unbinding to and from an asymmetric 1D track. And it's the asymmetry that's important here, as do cytoskeletal motor proteins. We have non-autonomous motion, i.e. that we provide an energy source from outside, which is chemical potentials due to changing the concentration of the ligands through external ligand pulses in a time periodic manner. Now, the point is what we've shown, but I haven't shown that, is TW can move against a rearward force, so it is a motor, but it detaches from the track at large enough rearward forces. So there's no stall or reversal of motion, which you would find with cytoskeletal motors. So, let's go on. Question, can we build a motor which mimics more aspects of protein motors, particularly kinesin? And that's where the aspect that I showed you of the zippering and the restricted binding comes in. So for my next protein, my next motor, I'll talk about what I call, well, you'll see the name when we get there. We've called this SCAM. It's not a SCAM, but it's a synthetic kinesin analog motor. Basically, it uses the same technology as the tumbleweed motor, which is why I introduced the tumbleweed motor, which involves repressor proteins, coiled coils, and peptide. So it's roughly the same idea. Here I have a schematic of it. So the actual motor has it has a repressor. These are the repressors now. I'm very sorry about that. So I better go to the next one. SCAM only uses 2 types of repressor, whereas TW uses 3. And the repressors I call A1, which is the same as A2, and B1, which is the same as B2. So I have one repressor at this end of the coiled coil, and an identical repressor at the other end of the coiled coil, and the distance between them is, the actual displacement between them is, I call D. That's 5 to 10 nanometers. The importance here is that I have another 2 repressors, which I call B2 and B1, and I have the same distance between them, but when I join A2 and B2, I have to make sure that to get the zipper effect I want, that these two are joined by a coiled coil. I call the distance E, which is less than D. I usually use E as a half of D. But the track that I have, which I've labeled, I better go on after this, that you have the DNA binding sites, that's the track. So I've labeled those A1 and A2, they're a distance D, B1 and B2, because they have to correspond to binding of the machine up here, which I hope is the machine. So I have A1, A2, B1, B2, A1, A2, B1, B2. And the asymmetry is going to come simply through the fuel, actually, through the ligands. And what I have for the ligands is, you'll see in the next slide, pink dots of the ligands for the A repressors and blue dots for the B repressors. And recap, a repressor is a ligand-gated DNA binding protein. which we talked about before. Okay, so this is how the SCAM motor works. And there's an important point here. And the point is that I actually place the motor so that A1, it's here, so that A1 is, that A1 is on little a1, and repressor A1 is on binding site A1, repressor A2 is on binding site A2. I just have ligand A in the atmosphere. I don't have to have two ligands anymore, and these two are free to do what they want. So they can just move all over the place. I then, once I've done this placement, so it's a bit like the movie you saw, this placing, then what I do is I change the ligand pulse, and I don't take anything out, but I just introduce ligand B so that the two, so that you can have a binding of the B. Now the important thing is that what you find here is that because this is short, and that this distance is less than this distance, the only place that this can bind is here, just like kinesin. But I'll show you where the kinesin business comes in. So once I've got it bound like this in this stable state, for a long enough pulse for the A's and the B's, so if you like, the A continues, the B's introduced, then I remove the A, and I just have the B here. As soon as I have the B, these two come off. So as soon as they come off, this one's unbound. You get a very vulnerable, and I'll show you why it's vulnerable, configuration like this. But what's interesting is that as soon as you put that in, it can go all over the place, but the only place this can bind, because you have A's here, that can only bind to here. That's the only place it can bind. So what happens is, and that's the idea of the zipper, because that binds here, and in that way I shorten the motor. So this is the analogy to binding of neck linker onto kinesin motor head, just before binding onto the microtubule. So again, I'll repeat, just because I've released these two, I've got a B ligand in the atmosphere, and the only thing that can happen is this does a diffusional search and eventually binds here. Once that happens, then you go back to this situation, and as soon as you get this moving around a lot, until you introduce, you've only got the B ligands here. Until you introduce the A ligands, then you get the only thing that can bind, the only way of binding is that this can bind here. This one can't because that's too short, so you have restricted binding. So what you have here is that the previous step restricts diffusional search so that A1 can only bind to the little site A1, and then the cycle continues. So it keeps going forward because of the way you've pulsed the ligands. That's the actual, the asymmetry. As I say, pink dots are for A repressors, blue dots are for B repressors. These are the ligand dots. And in the end, you've actually moved 4 lattice spacings for the whole cycle. Okay. Let me just show you how this works. This is movie thanks to the work of Dave Lee, kindly volunteered his services. And it's a bit different here because I've introduced 2 other repressors to give you more of a zipper effect and to slow things down so that you can actually see what goes on. And there's a surprise coming. Oh, sorry, you don't see anything that way. Giving it all away. Okay. So we start that way, and you can see you have part of the zipper effect, and then you have the rest of the zipper effect, then you change the pulse, and it binds forward. Next you change the pulse again, so that the 2 blues unbind, and it zippers forward again and goes forward. That's the forward motion. Now suppose you cut the track, so it can't bind anymore after this. It can't bind forward after this. What's it going to do? Anybody guess? So suppose I just cut the track. So I'm here, it's really looking for the forward point in the track, but it can't find it. So it binds, but it can bind back. That takes much longer than if it could bind forward. So it binds back, and then it goes backwards. So in fact, this is a reversible motor. But I have to do it by cutting the track.
But you still have the probability?
Yes.
Pretty,
Oh, I still have the appropriate ligands. Oh yes, this is only ligand pulses. I mean, what I had first of all, I still have the same ligand pulses I had before, slightly modified. So I have the same ligand pulses. When it gets to the end, it's got the ligand pulse with just the B's, say. It can't find the forward B. I then introduce the A and B, and it binds. One of the things it can do is bind back, and then it goes back. Now, can we use this? This was, I know I went into Nancy's office and I said, I think we can get a shuttle. And she said, great idea, just cut the track. So it's amazing what happens when you collaborate. So this is a shuttle. This is actually with the right number of, this is the way it moves forward, basically. Now, when it comes to the end, you'll see that it goes, it sort of, first of all, it sits on its bottom first, because it can't find the binding site, and eventually it finds the backward binding site and goes backward. I'll explain this a little later. So now we've got a shuttle, so I treat both ends of the track the same way, so I can get it to go forward and backward. Well, that might interest, but that's exactly what happens in a neuron. Here I have a neuron, right up there, and you can see that I have a vesicle, and the vesicle has two motors. And what happens is, suppose this is full of neurotransmitters. That means the vesicle will go that way and deliver, and then it can come using one motor, using kinesin, which goes from the minus end to the plus end, and then once it's delivered, it can go back in some way and use dynein to return. So we could imagine this, but here I do exactly that, but with one motor, not two motors. And so one of the things that you could imagine is that when it gets to, we'll wait till it gets to the end, it has no load at present. When it gets to the end, it picks up a load which mustn't be too large, and then it takes the load to the other end, delivers it, and goes back. So in fact, this could be a very useful motor, I hope. So the next thing I want to talk about is a little bit about what happens when you try to pull the motor back. Just make sure that I'm not overdoing the time. I've still got a little bit of time. Oops, sorry. So what we have here is I'm talking about reversal and stall transition for SCAM in the case of non-ideal motion. I showed you ideal motion, but suppose, for example, I instead of just cutting the track, I actually put a force. I actually put a conservative force, a rearward force, which is pulling the motor backwards. This is a very important thing, because we want the motor to go forwards when I'm trying to pull it backwards, at least for some forces. What happens to this motor? Well, what can happen is if I, suppose I have the force, you'll have to imagine it, and I'm pulling it backwards, and at present this is bound this way, that's fine, and then I switch the ligand, so here I have ligand A, ligand B, everything binds beautifully, and then I take away ligand A. And so that unbinds. What can happen? Well, one thing can happen, of course, if we're lucky enough and the force isn't too big, it can go forward, i.e. you get the zippering motion forward here. And that's that. But if it's, as you have different first passage times, for example, under forces, it could be, there's a probability that it will reverse. As I showed you the reverse for the cutting of the track. So what you can get is you can get this binding to here. And then it continues and it reverses like I showed you. This is basically a motion which, when I change the pulse, when I change the pulse, unbind and rebind, it goes forwards. The pulses, I should say. Another thing it can do is we could also get A1 binding to A1, in which case it sits back, basically. I call that a temporary stall. Or else you could have A2 binding to A2. This one I've called temporary stall 1. This one I've called temporary stall 2. So let me repeat. I've got a possibility of forward, going forward. I have a possibility of reversing, and I have a possibility of it sitting, of the motor sitting back to what it was to start with, which I call stall 1. I have a second one called stall 2. So how do I handle all that? Well, let me just, I've already shown this movie, so I don't think I'll show it again. This movie was actually, I cheated, because the movie was under a force, but it did the same thing it does the same thing as if you were coming to the end of a track. So I don't think you want to see the movie again. So I'll proceed onwards. So, if you remember the various things that happen, here I have the probability of transition as a function of rearward force. So along here I have rearward forces which I have in piconewtons. And I want to say that 5 piconewtons, I forgot to say, 5 piconewtons was the force that stopped kinesin. So we are of that same order of magnitude here. Is there anything like a stall force? Not quite. So what I have is that if I put on a very small force, I should mention what the probabilities are. This is the probability of forward motion. I have a probability of forward motion with a small force, and nothing else happens at all. It can just go forward because the forward motion is very fast. But I'm slowly inhibiting the forward motion. So at something that I call a reversing force here, that's where the motor can start. It goes forward, but then at some point, it starts having a probability, this blue probability of reversing. Interestingly enough, also, as the probability of going forward of having a forward step goes down, the probability of having stalling, particularly the, stall number 1, really rises. So it can easily sit back, and also stall number 2 isn't so big, but that also rises. The point is that there's always, you start having a finite probability of it reversing. As soon as you get that one reversing step, it'll continue. It'll start going backwards nicely and processively. So what you find is, just as I showed with the motor, that it can come to the end, it can stall, but then eventually, when I get the probability of reversing, it'll reverse. So I don't want you to bother with the temporary stalls. I just want you to have a look at the forward probability and the reverse probability. And what we do is we arbitrarily call a stall force a force where the probability of going forward is the same as the probability of reversing. But in this region here, you get forward motion, which is followed by reverse motion, and you get some intermediate stalls. Then finally, as you increase the force, here I should mention this is a very short Ligand pulse, but I'll show you what happens as a function of Ligand pulse in a moment. So concentrate on this reversing force and on the stall force, but eventually what happens is that the machine can't go forward at all. When the force is strong enough, it can only go processively backwards, but it can stall. So what you find when you look at, I haven't time to show you, you find that when you pull it, it'll start by stalling, may go one step forward or something. It'll start by stalling either here or here, and then eventually there'll be a probability of reversing, it'll go back. So you have 3 regions. You have pure forward motion, you have pure reverse motion, and then you have this intermediate motion. So I've called this a stall region. If you actually have a molecular motor like kinesin, you can put a force on it that will stall it, and then it'll go backwards. Here we don't have a clear stall, but it behaves more or less the same way, but in using a different mechanism. Okay, so I then want to just show you this. What happens is, here I have this is rearward force in this direction, and this is pulse time. So up to here I have 0.6 milliseconds for the Ligand pulse time. I'm assuming all the pulses that I showed you, the 4 different pulses are all the same length. And what I find is, first of all, the reversing force. That's the force at which you start getting reverse motion. You can see what happens. That increases. Initially, you have a region here where at very, very small Ligand pulse times, you can actually get reverses because the pulse is too short. As soon as the pulse is reasonable, you get the picture that I just showed you, and you have a reversing force, which increases. And I think eventually will saturate, but I haven't really simulated that far yet. The stall force is, of course, larger, but behaves in the same way. So as you pull more strongly, as the Ligand pulse increases, you can imagine that the stall force increases, but you have to get to a point where it just flattens out. It doesn't matter anymore. So, I'm sorry?
I mean, the previous falls get probably add up to 1.
I see. Oh, the probability doesn't add up to 1 because you've got the other possibilities of it stalling. They should add up to 1. They did add up to 1 when I drew it. So that should actually technically add up to 1. If it doesn't add up to 1, then I haven't plotted it properly. But certainly in the simulations that I had, everything added quite nicely up to 1. Because here it certainly adds up to 1, and I think if you add these, because 0 is here, I mean, I can show you the numbers. They do actually add up to 1. You may not be satisfied with this answer, but they do add up to 1. I have all the numbers, and in fact, they should do. I might have made some mistake in plotting. But certainly, this is the way it goes. I've plotted a lot of these, and they add up to 1. Okay. Let me give a summary. So SCAM is able to perform work against an external force. Sorry about that if they don't add up. SCAM can move forward processively for rearward forces less than a reversing force, which depends on the ligand pulse time. A quantitative measure of motor behavior is the average thermodynamic efficiency, and we have a very small, we have a sort of intuitive way of looking at it in terms of chemical potentials, because it's really the energy that's taken out when you bind and when you unbind, and that is chemical potential gives us something that's quite large, 12% to 36%, but we have to look at that again. We haven't really done it properly, but this gives you a rough idea. Kinesin is 50% and maybe more. The SCAM speed depends on ligand pulse time. It's completely controlled by ligand pulse time, but as I showed you, that as I mentioned to you, that if you actually go to very, very small pulse times, you can the pulse times are not long enough, and you get reversals, and you get the motor coming off. And so I do have a minimum value for the numbers that I used, which is about 0.35 millimeters per second for an ultra-low pulse time of 30 micrometers per second, 30 microseconds. And this is extremely small. You'd never get a pulse time like that. But that's just a lower limit. Okay, just to finish. And so what we have is SCAM mimics kinesin neck linker zippering, followed by restricted binding site search and binding to attract. So it really mimics in a very controllable manner the way that kinesin works, because we're powering it from outside, but we don't have a power stroke. So SCAM can be used as a transporting shuttle, which is a very useful property. I just want to say, thanks to talking with various people, Mike Kirkness and Dipankar, that changing the ligand atmosphere is a bit awkward. You know, there is, that has been constructed and can be done. But if you can change the ligand-gated binding protein to a photo-switchable one, so instead of putting, making it change conformation and bind when it binds a ligand, you might be able to make it change conformation with a photo switch when you put on a specific type of light, i.e. blue light, for example. This one works the other way. In this case, with this particular one here, it's photo switchable, and what happens is it binds in this form. In the absence of light, you put on light, it changes conformation completely. Don't forget it's attached to a hub in our case. So these don't come apart. And it changes very quickly, I believe. And then when you take off the light, it can, you can have a, you'll have a sort of thermal diffusion where things start coming to, or you'll have a thermal behavior. It'll be temperature fluctuations will work to make it come back to this. So and that might be rather slow. But at least you can take your whole apparatus, you can bathe it in light, and then you can switch the light off. So that's, I think, an improvement. So thanks to everybody for these suggestions. I'm not going to show anything to do with this, but we have another motor. This is a DNA moving in a microchannel, which is coated with repressor proteins, and we use salt to contract it. I'll show it very quickly. I can't. So it's sort of. But I won't explain it. You'll have to ask questions about it. So that one does have a power stroke, which is induced by salt and electrostatic interactions. So I have to stop that. I don't want to talk about that. Then we have Nancy Ford talks very nicely about the lawnmower. This is actually a motor which behaves like a burnt bridges motor on collagen. It actually cuts up its track and hopefully moves forward. And we have our two experts here, Susanna Kovacic and Lale Sami, who's in the audience, who are actually building this. But if you want to ask questions, they're right here. So I wish to thank all the wonderful people with whom I've had the opportunity to work on this project. Special thanks to the motor group I showed you, the Human Frontier Science Project motor group, the Ford Lab, the Jennifer Thewalk Lab, the Friskin Lab, Chi Ming, who's in the audience, and found my mistake, and Dipankar Sen, Dave Lee for the movies, and Adam Waters for the movies. And here you have a picture of my marvelous family. These are only 5 of the one child and 5 of the grandchildren out of 14. Thank you very much.
Martin? So, questions, please.
Please. Oh.
It was very clear now, Martin. I mean, having spoken with you, I mean, it wasn't as clear as what you just described. I mean, when I spoke with you before, it was never as clear as how the beautiful presentation.
I'm thankful for that.
But I was curious that your simulations with the two-legged tumbleweed, or whether it's a tumbleweed or is going in some other way.
The tumbleweed is three-legged, and this one's the other one's more or less two-legged.
But your simulation is a two-legged entity,
yes,
whichever way it moves.
Yes.
So what is the appeal of working experimentally with the three-legged creature? Because the chance of something going wrong is probably to the power of n, of number of legs to the power of n.
Well,
so,
I think that, oh, you mean why work with the three-legged one?
With the two-legged experimental system.
Well, the two-legged was sort of discovered way after the three-legged was started, people started working on the three-legged. So what happened was that, what happened, the history of this is the first motor we looked at was the tumbleweed. That was actually a motor thought up by Paul Kermey, who's in Australia. And I simulated it, and also a very good student working with Heiner Linke, Nathan Kawada, simulated it. We did a lot of tuning, finding out what interactions were supposed to be, and the next step was that the motor would be constructed. Now, the motor, the track was constructed here by Lale, I think, and by No, who's gone, and that's the track, the DNA track. The motor was constructed in Australia, but the problem was that 2 legs were connected by Intine chemistry, but it was found very difficult to connect the 3rd leg.
That's why it's probably best to stay with the 2 legs.
Yes. And no, the 3 leg, the 2 legs, it has the same problem. You still have, you still, in the middle, you still have to join 2 of them and have a 3rd. So you have a real problem there. But the point is that until that was done, I went to Australia again because I have grandchildren there who were unfortunately not shown. And when I went there, we started studying kinesin. We looked at the papers, and I spoke 1 hour a day to Paul, and we were thinking about how we could use it. And so that's when after a couple of sleepless nights, we thought the motor out. So in fact, the second motor perhaps should have been looked at first, but it's way after. And that may answer your question as to why it's not.
Just a small comment there, and to physicists it may not be interesting, but you can get 2, just by using this system of repressors and repressor binding sites, you could have a motion on the DNA which is literally going on one face of the DNA, or you can have a precessive one where it follows the helical track.
Yes.
So it goes around and around. So those 2 kinds of motion.
But one would rather have it on top,
but,
I guess.
But, yeah.
And what we've tried to do is we want to the motor is actually just a little above a sort of a lipid bilayer, So hopefully it's not going to be able to go underneath. I should mention there are lots of things.
You can position the things at will and make it go that way.
Or you can make it go that way. That's what I'll have to talk to you, because that would be a very good way of doing it. I just wanted to say that there are other things I haven't included, such as non-specific binding, which is weaker, but we do have that in the paper that can cause some problems.
There's another question.
Yeah, a very enlightening presentation. Thank you.
Thank you very
I have
much.
two questions. One, you showed a animation of the DNA strand moving on the microfluidic channel.
Yes?
What are the dimensions of those channels?
Okay, I can answer that conveniently. What happens is that you have lambda DNA in there, which is roughly 16 micrometers long. But the point is that the persistence length is, i.e. the effective bond length is 100 nanometers. So what you do is you have this has already been done. You have a microchannel of the order of 50, 100, 200, and then you put your DNA in there and it spreads out because it can't reverse. So that's the main point. Then what you do I love talking about this one. Then what you do is you have it in there, and you actually coat the microchannel with repressors, and then you start the pulses. But you again have the same pulses that I showed you for the SCAM, but you also have salt pulses. So the salt pulses, if you have high salt, it'll contract. If you have low salt, it'll expand. So if you work it rightly, you'll get it moving like this. That's the way it works.
My next question is, the bacteria have the flagellar motor.
Yes.
How does this compare with that?
Badly, I think.
Oh.
No, this is. I think what we're trying to do here is, I think it would be very, very hard if we tried anything with rotary motors, because you have the flagellar motors and you have the F0, F1 ATPase motor, which rotate. I think these are they're multi-components, and we have nowhere near those components. I mean, that's why we stuck to the cytoskeletal motor, because they have two legs, and that makes life simple. We have other motors that look like that could be used for that. But basically, for the inchworm, the scam, and the tumbleweed, those three, we base things on the motors, because they're sort of, in some sense, conceptually easy to deal with.
So these
with few components.
Translate back and forth.
Yes.
Okay. Thank you.
But if you look at the other motor, which was the motor that I showed you, the lawnmower, which eats up its track, that's completely autonomous. But it does have problems because you eat up your track. That's really like having a truck which is braking the road when it's moving, braking the road hopefully behind it when it's moving. But it might break the road in front of it. So you have a bit of a problem.
I have one more question.
Oh, absolutely.
Is there a lifetime for this, or so long as you give ATP, it'll keep going forever, or is there a lifetime?
You mean? Well, I think at some time. Well, what happens is that you mean the kinesin motors?
The whole structure.
For the kinesin motor, for our motors, I don't know. But for the kinesin motors, it has to get from one end of the track to the other. So the idea is it has to be quite processive. And as I said, it can go 100 steps, each step 8 nanometers. Before it comes off. So basically, and that's what you mean by a processive motor. I happen to show you my nice little motor, the SCAM, which is completely processive in what I do. But when I start putting non-specific binding in, then I find that it's no longer processive, it can come off. So it's no longer perfectly processive. And also, the other thing I haven't included, which you do include in the case of kinesin, is that I've said, it can bind, the motor can bind, it binds immediately. It's not the motor, but the repressor binds immediately at one point, as soon as you put the pulse in, and then it comes off when the pulse stops. But that's not good enough. You do have relaxation times, and so one has to go to a better form in order to get the reality for this, which we've done for the tumbleweed. You actually have to use something like a master equation or a continuous time random walk, which is a bit more difficult. Yes?
I enjoyed it a lot, Martin.
Very much.
The thing about kinesin is that it doesn't require pulses, because one ligand, the blue ligand maybe, turns into the pink ligand through hydrolysis.
Absolutely.
And so you could accomplish the same thing here if you only gave it pulses of blue ligand and gave it an expiry date, so it turned into a pink ligand,
Oh, yes,
and
I
then
agree.
just had to
Yeah.
reintroduce the blue ligand over again.
So you'd have to do some chemistry
Yeah.
to be able to do that.
But then you would not have fixed pulse times.
No.
So if you randomize your pulse times, will this still work efficiently? And is there a kind of will that affect the efficiency with which it can turn around? For
I
instance?
think the answer is how random do you have the pulse time? If you can, the pulse time has to be a certain minimum, and if it's greater than that minimum, then your
stall
it'll stall.
Will
And then, of
more-
course, that minimum has to increase if you put a load on it. And I did forget to mention that one of the simulations I want to do is to put a load on SCAM and see a proper load, not just a backward force, in which I have a stalk. That's going to be harder and a load. You do have that problem.
Because then if you're only giving it the blue and waiting for it to convert spontaneously and randomly to pink, then the problem is that there is some time where you're not giving it any ligand at all. You're just giving it blue and then nothing, and then it can fall off.
Yes, well, I think what you'll find is you'll have to work it properly so that you get a good processivity like kinesin. And then that's a very good, nice idea. I have to talk to you more about that. Thank you.
If not, let us thank.
Thank you very
Martin
much.
again.
I wanted.
Thank you, Martin.
I want to say one thing more. I really want to thank Veso and Pam and all the staff of IRMACS, the staff of IRMACS, because they're the ones who did the hard work for this Royal Society series. I was just the guy who thought it up and mentioned it to Veso. And then Veso, and Pam, who are amazing people, got it all going. So I'd really like you to applaud them.
Oh, thank you, Martin.